Hello, everyone. This is Miss Dagenford, and I'm going to try again because it's already quit on me a couple times. So we'll see. So tonight we want to look at enzymes a little bit more and also prep for the lab. Our goals for the video would be to make sure we understand how enzymes work, why they work, and what happens to them when we give them some challenging conditions. So let's go ahead and start. Enzymes are proteins, and I put mostly in parentheses because there are a few exceptions, but they're pretty rare. Um, by and large, when we see an enzyme, that's a type of protein. Now, we talked about in class that enzymes are and proteins are made of amino acids. So we had that breakdown of the nitrogen and the carbon, and you can look back in your notes if you just want to confirm this. You don't have to right now, and they were distinguished by this side chain. So here's one amino acid, and then if we connect them to another amino acid, it's another um, nitrogen, carbon, carbon, and then so forth and so on. Well, as they connect to each other, I showed this today in class. They can bend around because they're attracted to each other's positive and negative charges. Now, if I extend this and make this smaller, we get a ribbon of these amino acids. And as they fold and turn and twist, they create this elaborate three-dimensional shape, and it's held together because of those bonds. Now, if I keep doing this, you're going to have some areas where you've got the polar sections that have the partial charge, and then you have a non-polar section. And just like a ball of yarn, and if you were to look at a ball of yarn, you would say, hey, it looks like a ball. But if you looked at it more intently, you would see that it's made of yarn. Well, that's the idea. If you were to look at an enzyme, you'd see this chain of amino acids, but you would see that it has an overall shape to it. And that shape is going to be very important. So, oftentimes in books, you just notice that the outline is given, and sometimes they shade it in to kind of give it a solid shape. So you're going to hear me say that a lot, and that's going to be very important. That shape to an enzyme, and indeed the shape of any protein, ends up being very crucial. Now, why that is ends up being crucial to how an enzyme works. Well, let's take a step back and remember that reactions can only occur if they can get past this activation energy. So, this activation energy is usually pretty high. Most molecules, and here we can draw a little disaccharide. It's got a double ring structure. And these disaccharides don't just spontaneously split apart. Paper doesn't spontaneously combust. It has to have a certain amount of energy, a certain amount of heat, a certain amount of transfer of energy to get it up to that activation energy. Now, once we did that, it would burn. We could take a spoonful of sucrose, spoonful of sugar, put it over a flame, and that would burn. And then that energy has gotten it over that height of the activation energy. We've given it energy. Now, if you were to lower the activation energy, enough molecules might have that energy to get over to the other side. And let's say the products in this case are two monosaccharides. And so let's say that, I'm breaking them apart. Well, this doesn't usually happen. You don't usually have a reduction in the activation energy alone. However, enzymes can very nicely work to reduce the activation energy. So the smaller hill is the activation energy with enzymes. And that's why enzymes work. They reduce the activation energy, and that's going to increase the rate of the reactions happening. Just like if an amusement park lowered the price of admission, a lot more people are going to be able to go through those gates and get to the other side. Well, if I reduce the amount of energy, I'm going to be able to get to the other side of the reaction. So we increase the rate of reaction, and that happens because that activation energy has been reduced. So let's look at how this happened, the mechanism. Well, and you do not have to draw these exactly as I've drawn them. The idea is that the shape is important. So let's take a look at an enzyme. Let me for a second go ahead and erase that. This little orange pocket, and I'm going to highlight it. This little orange pocket right here, that is called an active site. And that active site is going to be highly, highly crucial because this is where a molecule that fits that active site is going to bind. So you have a molecule here and it fits that active site quite well. Now enzymes are highly specific. So we could have enzyme A in the orange, and then just to have a little fun, I'll change colors. Let's do a purple one over here. Now enzymes, and again, you don't have to try to make yours look exactly like mine. I don't like that for some reason. Okay, so that enzyme has to be, so let's just make it obviously different. So enzymes just have different shapes depending on what substrate. And so enzyme A has a substrate like the blue one. Enzyme B is going to have two substrates like this. So let me be clear. The red and the blue, those would be substrates. And a substrate is what the enzyme acts upon. It's going to either break something down or build something up. So in the case of enzyme A, it might very well be breaking that substrate down into two molecules. On the other hand, over here, the substrates might be joined together. So after the reaction, we might have a molecule that is now one. So in the reaction A, we're taking one molecule and breaking it apart into two. With enzyme B, it's taking another two molecules and connecting them together. They were two separate. We create a bond together and we make them one. And I can give you an example of how that happens. Over on the left with A, this is going to be digestion, right? We're going to take some food that we eat. Let's say we're drinking milk and this is lactose, and enzymes break apart that bond and we're breaking down our milk that we eat. Over on the right, this might be when you build muscle. When you build muscle, you've got to connect together um, molecules to make protein. We're going to connect amino acids together to make muscle. So in B, we're building. In A, we're breaking down. But enzymes can do both of those things. Now, that active site is going to form a very temporary bond. It just holds it. These could be those hydrogen bonds. It could be ionic bonds. It's going to hold it in a very, very temporary attraction. And when it does that, it's then going to change its shape ever so slightly. And as it changes the shape, it's going to either break a bond, it's going to, just like you grabbing a cracker and bending that cracker until it breaks, that enzyme could put stress on those chemical bonds and then the chemical bonds break. Or you're going to hold those substrate molecules in such a way that they join together. I like the way of thinking about Legos. When you're putting Legos together, those Legos can only come together if they're held in the proper position. And if they are presented in that way, they can then connect together. Well, that's the same idea of the lock and key. The lock and key is going to create a proper orientation. So here's our enzyme again, and we're going to then hold our substrates together in such a way that they're going to bond. Or it's going to put tension on the bonds, and then that's going to break. So here's an example of what they can actually look like. Now, it doesn't look like my drawings, right? Well, again, all of the little circles are amino acids, and how they get these cartoonish shapes is you look at that outline and you kind of draw around it, and you can imagine when they're making these diagrams, they want to simplify it. So we have a shape. Here's our enzyme, and then here's our active site that's going to allow the substrate to bind to it. Now, you want to be thinking, if that active site, if that shape is so important, well, what happens if that shape is gone? Um, and remember, the shape is coming from that three-dimensionality of that set of amino acids that are bending and folding and creating all these twists and turns that ultimately create this little pocket. Now, you want to think about the fact that if that starts unraveling, if that loosens up, then we no longer have that pocket. So if we were to unravel this, then we've lost our active site. So over here, let's say that our active site is right here. Well, where is it over on the right if we've unraveled it? And in our lab, the next day, we're going to take a look at that. And what we're going to be doing is setting up test tubes with an enzyme called catalase. Now, catalase, and you'll learn that anything in an ACE is an enzyme. Catalase breaks hydrogen peroxide, which is a harmful chemical, but catalase is going to break hydrogen peroxide into water and oxygen. And I'm hoping you look at that and notice that this is not balanced. Let you have a little bit of fun and you can balance that. We'll talk about that on Wednesday. But what we want to do is examine whether or not the enzyme is working. And the way that we're going to know whether or not the enzyme is working is if we see oxygen. Because if we see oxygen, we know that hydrogen peroxide has been broken down. Now, I want you to read the lab. I want you to put the lab in your lab notebook. And I want you to be familiar with the processes. And I want you to really, really hone in on that fact of, if this active site no longer exists, then the substrates can't bond to it. And if the substrates can't bond to it, you're not going to see the reaction go through. And to go back to ours, if there's no oxygen being produced, then we know that the reaction didn't happen. Well, I hope that gives you a little bit of help understanding enzymes and how they work. We'll talk about that a little bit on Wednesday. Have a lovely day.